Thanks everyone for coming. My name is Steve Brady. Really happy to be here. I suppose I have to stand back here, right, for the filming and all that? Yeah, probably. I will. So I'm going to talk about a couple different amphibian species today, primarily the wood frog, which if you squint is sitting there at the bar having its dreams dashed by the presence of roads on the landscape. And I like this photo. So this was taken a couple months ago during the eclipse. I was the lucky person on the highway, on a long stretch of highway in Vermont, driving northbound after the solar eclipse when everyone was driving southbound. And it occurred to me that this is very similar or emblematic of my study system, where the same road can have very different consequences depending on who you are. So I was the lucky one, and I'll see if you kind of pick out the two species, who's the lucky and the less lucky here. So I'll talk about roads today, their source of pollution, their presence on the landscape. In the US, they say roads take up about 1% of the surface area of the country, but about 20% of the landscape is affected by their presence. And effects include, not surprisingly, edge effects, roadkill, invasive species that are introduced through roads, and then lots and lots of different pollutants coming off the road from the road surface itself. And surely in Montreal, this is a familiar site. This was taken in the Adirondacks many years ago. But if you haven't seen this, this is a salt truck that's plowing, removing snow, but also adding salt to the road to melt the snow and ice. I like this video because the truck drives over a bridge right here, and the bridge is graded, so the salt goes right through into the river. So not even run off, but run in. And there's a lot of salt. So over the years, salt used for de-icing has become the most prevalent use of industrial salt in the world. And what that means for fresh waters is that the amount of chloride in these surface and groundwaters is going up and up. And we see that in rivers, we see it in lakes, we see it in streams, we see it in groundwater even. So for me, thinking about this many years ago, I started to wonder, well, are populations adapting to roads, their presence, their effects, and road salt in particular, because where I started doing this work, road salt is one of the big contaminants that we find. So I asked this question for a couple different species, so the wood frog on the left and the spotted salamander on the right. And my approach was to look at a series of populations that I would describe as woodland that were located far from the road, at least a couple hundred meters away, where we found no evidence of road salt, and those that were within 10, 15 meters of a road, or roadside populations, where we saw a lot of salt. And of course, there are other pollutants in those ponds as well. This is what those habitats look like. So on the left is one of my favorite pools in Connecticut. It's named after my girlfriend, but she's not here, so I might change the name later. No, but it just typifies a beautiful pool in the woods. They're small, shallow, vernal pools. And on the right, a really similar looking pool, but this one happens to be next to a road. And the difference between these two in terms of salt is really dramatic. So what we're seeing, we've just put together a data set across New England looking at salt in these roadside ponds, and we find that they can be elevated up to levels that we would expect to find in brackish environments. So we see distance within, here's just one statistic from this data set, 70% of these seasonal vernal pools within 37 meters of a road exceed the federal chloride criteria set forth by Canada, for example. And we also find this figure on the right shows us that there's stratification in these ponds. So we've seen stratification in lakes that get salt, but in these small ponds that are only 3 feet deep or less, we're finding really strong stratification patterns that persist throughout the year, which have big consequences for things like amphibians that will migrate up and down in the water column for food and to avoid predators. And this, the video isn't playing, but this is a typical spring rainy day when these animals are breeding. So they come out in mass in the spring, lay their eggs, and at the same time, these warm spring rains are washing a lot of salt and other pollutants from the roads into these habitats. So to think about some population level differences, one of the first experiments I ever ran many years ago showed this kind of classic signature of local adaptation for the spotted salamander species in these habitats. So we studied 10 different ponds, did pairwise reciprocal transplant experiments, and found that roadside ponds on the right were not great places to live, but if you came from a roadside population, the red line, you had an advantage. So we saw this nice evidence of local adaptation. And then we repeated this experiment for the other, one of the other amphibians living in these habitats, the wood frog, And wood frogs are pictured here. This is the little black tadpole swimming around. And in the background is a spotted salamander egg mass. So these two species live in these same ponds at roughly the same times of year, undergoing similar periods of development and metamorphosis at about the same times. And so you might expect that they would show similar patterns, but they did not. So now the red lines in this case, which are the roadside populations of wood frogs, are always below the blue lines, showing that they have disadvantages in these environments. And kind of curiously, roadside populations have disadvantages not only in the polluted roadside environment, but also in the woodland environment when we transplant them out. And we've done this many times and found these results most of the time. This pair of slides is contrasting the first set of experiments where we just took eggs out of the water, and then another set of experiments where we thought, oh, maybe those first 2 days in the water compromised their survival. So we did a controlled breeding and we found the same result. We found this sort of maladaptive pattern, again, red line below the blue line, for these roadside populations in the context of salt exposure experiments. So here, lower survival with acute exposure to road salt, and increased prevalence of malformations in those roadside populations when exposed to road salt. And so some of the things we wanted to follow up with over the years when we saw these patterns was whether those survival detriments extended all the way to metamorphosis. So most of these experiments are from back in my graduate school days, where I would stop the experiment when the animals finished hatching or somewhere later down the line, but not all the way to metamorphosis. I don't know why, that's just what I was advised to do. But getting back to the system, when I had students of my own, I said, well, we're gonna follow up on this and run this experiment all the way out to metamorphosis. And a student of mine, Mia Forgione, did that and found that, yep, even if you run this experiment out to metamorphosis, we still see roadside populations have lower survival rates in the context of road salt exposure. And Mia was clever and thought to look at different developmental stages to see if there were any signs of stage-specific sensitivity. And sure enough, it seemed to really be embryo-specific. So those embryos among these different stages, embryos, hatchlings, and larvae, seem to be most sensitive or differentially sensitive compared to those woodland populations. And Mia also found that it takes these animals longer to metamorphose in these ponds, and she postulated that would kind of cause a double whammy effect where you're slower to develop and you spend more time in the pond exposed to more pollutants. And then we contrasted this with some of that earlier work I showed you and found that, yeah, in those field experiments too, embryos had that survival disadvantage, but not the larvae. So there seems to be something going on where these roadside populations are more susceptible to the effects of pollutants in their ponds, and that seems to be stage-specific, something occurring early on in embryo development. Recently, another student of mine found that, oh, there's no found nothing, just a nice, they all disappeared. So if the data were there, you'd see that the roadside populations are more sensitive to the combination of salt and temperature. So again, she found roadside populations did not like salt. They preferred it less than woodland populations, but that effect was exacerbated for animals in warmer temperatures. So we see there's some interaction with thermal environment there. And just getting back to these embryos a little bit, the cluster of eggs on the left are wood frog eggs from a roadside pond, and the ones on the right are from a woodland pond. And some of that is just differences in tannins, the difference in color there. But one of the things that has struck us is that those embryos are potentially different sizes. In the roadside environment, the mass is more condensed. And that's something we've looked at recently, is how this osmotic difference in the environment might contribute to what's going on with these embryos. And so we have just a little bit of time-lapse imagery here that might play, if we're lucky, showing the development of two embryos. One is in a little road salt bath on the left, and one is in tap water on the right. And over time, what you can see, and I'll fast forward here in a minute, is that the embryo in the salty water, its vitelline membranes that encase the embryo don't fill up with as much fluid. And so some of the malformations that we've seen in the past appear to be just perhaps a product of a spatial limitation, or like a spatial constraint in these eggs. Let's see if I can get to something here for you. No? I'm trying. well, that's a bummer. Okay, it didn't work, but I can show you later. So at the end of that video, what you'd see is the embryo on the left, its membranes are like this big, and the embryo on the right, its membranes are twice that size, and it prevents the embryo from actually elongating. So we see these embryos are actually, as they develop, they become kind of curled up around themselves, and they don't have this space to stretch out. So we think there's something going on at the embryonic stage, and some of it may be related to this osmotic difference creating a smaller encasement for these animals to develop in. These effects I'm going to talk about next aren't population specific, or rather it's not a divergent response, but we do see, I wanted to point out that we see this strong edema response in roadside wood frogs. So the animal on the left is a typical wood frog during breeding season. The one on the right is the most severe that we see this, but these animals can get really edematous. And we find that edema in these ponds is linked to the amount of salt in those ponds. So this figure on the right, top right, is showing conductivity, which is a good proxy for road salt, and the prevalence of edema. And we see that it also has a negative association with jumping performance. Not surprisingly, the more edematous the animal, the less well they jump. And just a quick picture, you can see there's another bloated frog on the left, and we did some work to aspirate that edema and see how that affects their swimming performance and their jumping performance. And if you relieve the edema, they swim and jump much better. And you can see that geyser of fluid coming out. So some of these animals are actually carrying up to 50% of their body mass as excess fluid at the time of breeding, which is a really critical time. Okay, so lots and lots of negative effects out there. And yet, when we look at populations in roadside environments versus woodland environments, we see that they're generally there. They haven't, in most cases, gone locally extinct. Population sizes tend to be about the same when we scale them to the area of these ponds. And so, of course, it prompts the questions, well, what's going on? My first thought before thinking about the population effect was, well, these animals are becoming maladapted. There's something evolutionarily degrading them over time. But we still see their populations persisting. And so I've kind of shifted more towards a potential like trade-off scenario. And sure enough, we do see trade-offs at other life history stages. So fecundity, for example, here's female body size on the x-axis and eggs per female on the y-axis. And you can see the slope for roadside animals with the dark points there is steeper than the woodland ones. So we have higher fecundity for females in these roadside environments. We see some benefits to locomotion for females in those roadside environments. So they jump further when gravid on their way into the ponds than females from woodland ponds. And then recently, this is, I guess, what I'm most curious to get feedback on while I'm here, we've started delving into some of the gene expression and alternative splicing patterns as a hopeful future direction. And this is a collaboration with Jintao Hu and his lab at Fudan University, and Jintao's at the meeting this year. So if you see him, you can ask him questions about this. But what we've seen is that just generally, we see really strong differences in gene expression between life history stages of hatchlings or larvae and adults. So that's regardless of where these animals are coming from. And we see that for both gene expression and alternative splicing, which is exciting for us to think about that. When we look at the larvae, we find that there are differences in gene expression. And so I think we found not a tremendous number of differentially expressed genes, but what was intriguing to me is that they clustered by populations, but they didn't cluster by population type. So we didn't see any striking patterns of roadside populations versus woodland populations in terms of their gene expression profiles. We did find two, and I thought this was exciting, but I guess it's not that many, two divergently expressed genes that were parallel. And one was a G protein signaling modulator, and that was upregulated in roadside versus woodland populations. And without knowing anything about these two genes, that and the heat shock protein, when we looked in the literature, it seemed that the signaling modulator was generally associated with cell growth, often in a negative way, so with tumors and uncontrolled cellular growth. And heat shock protein, we all sort of expected that it would be upregulated in the stressful environment, and it's downregulated. So it maybe maps out with what we'd expect for these sort of maladaptive responses, but we're not quite sure exactly what to make of all this yet. And again, we looked at alternative splicing and found kind of mirrored patterns with what we saw with gene expression, where differences between larvae and adults, but nothing striking for population differences. And I think that's time.
Hi, everyone. Thanks for coming out to talk. I'm Andrew Whitehead, and I'll be talking about some of the work that we've been doing recently on the genetic architecture of evolved pollution resistance in multiple populations of fungulus killifish. So as we all know, the Anthropocene can changes the environments in swift and severe ways, and that can impose pretty severe fitness challenges on lots of different creatures. And those fitness challenges can come from lots of different dimensions of environmental change. One of the major research themes in our lab is the pollution dimension. And when we think about toxicants, we think about toxicology, we're faced with what I like to call this sort of tragic irony of evolutionary toxicology, that the creatures that we want to kill with chemicals tend to evolve resistance, and the creatures that we don't intend to kill with chemicals tend to decline towards extinction. And you can probably notice some differences between these kinds of creatures here. And so some of the factors that might predict these two different kinds of outcomes would be population size and generation time and the nature or complexity of the adaptive challenge. So for these creatures, massive population sizes tend to be pretty fast generation times. And I would argue that when we're talking about insecticides or antibiotics, we're talking about fairly simple adaptive challenges, like single chemicals that are guided cruise missiles. They're designed to kill through very specific receptors, right? Whereas in this scenario here, these creatures tend to have small population sizes, relatively long generation times, and they encounter pollution, which tends to be a complex mixture of chemicals that weren't designed to kill, but can be toxic nonetheless and perturb multiple biological systems that might be stage-specific or really diverse. So we think of the cruise missile scenario in the first and the cluster bomb scenario in the second. Much more complex adaptive challenge for pollution compared to agricultural or biomedical scenarios. What also contributes to the complexity of the adaptive challenge or ability to predict perhaps the likelihood of adaptive outcomes are the genetic architectures of the traits that might be relevant in these scenarios. How many loci are involved? What are the effect sizes of these loci? How might they interact? And what kind of biochemical pathways do we find them in? So work in my group over the last few years has been focusing on a creature that arguably fits the lower scenario. It's a vertebrate creature that's evolved resistance to pollution, which appears to be somewhat rare. Maybe we're just not looking for pollution resistance, but agricultural pesticide resistance and biomedical resistance reaches out and punches us in the face. Or maybe it's just rare because it's hard. But killifish are total weirdos here. So they're this big. They're about the size of your thumb, and they're the most abundant thing with a backbone in salt marsh estuaries on the East Coast, the Gulf Coast. They also tend to be abundant, if there's otherwise suitable habitat, in urban estuaries. So we have been studying with a number of colleagues 4 pairs of populations, where the pairs are geographically nearby clean site, polluted site populations. We got 4 pairs, and this is the color scheme I'm going to be following. The color indicates a geographic pair. A pastel color indicates the clean site. The bold color is the polluted site. Okay, so 4 pairs of populations. And here's a model toxicant. So these estuaries tend to be polluted with the forever chemicals of the 1960s and 70s and 80s, so PCBs, dioxins, PAHs, also a bunch of heavy metals. I mean, these are complex, highly disturbed habitats. We take a model toxicant here, a PCB. We look at survivorship curves, and here are the survivorship curves of normal killifish populations from clean sites. They're pretty sensitive, very sort of narrow dose range over which they don't survive. And here are the survivorship curves of their geographically nearby pollution site population. These are F2 laboratory raised common garden experiments. This is heritable. And I should point out, if you haven't noticed already, that's a log scale along the x-axis. This is not a subtle phenotype, right? Some of these fish are can survive what's normally up to 8,000 times the normally lethal dose of these chemicals. So this isn't a subtle phenotype, right? It's a pretty extreme resistance phenotype. And when you look at sort of genetic relatedness of populations, or geographic pairs sort of pop out as most closely related to each other than to anyone else. So this is sort of consistent with this idea that this evolved resistance has evolved repeatedly, independently, multiple times. We also see this in a Gulf Coast sister species in the Houston Ship Channel that's evolved resistance independently. And you take sediment cores from these sites and you can reconstruct the history of pollution, the history of PCB accumulation. And we're talking like it started accumulating in the 1950s, peaking in the 70s, was banned in the 70s, and so there's been some recovery since then. But this tells us that the evolved resistance, or at least the selection pressure, has been there only very recently, and these creatures are about 2-year generation time in the field. So we're talking like a few dozen generations to go from 0 to 60, massive adaptive leap. So how do you do that? How do you go from 0 to 60 in a few dozen generations? What we started with was sort of the genome scan approach. Where in this plot here, we've got the pollution site genetic makeup compared to the nearby normal sensitive population genetic makeup, and we're looking at signatures of selection as we scan across the genome, alternating black and gray or different chromosomes. And we've highlighted in red are these footprints of natural selection, right? And we can see this for all of our 4 resistant populations. Compared to their nearby reference population, we see lots of loci showing these big fat signatures of recent natural selection, these big selective sweeps. But conspicuously, there are very few loci that are shared signatures of selection across these populations. Now, there are a few that are shared, and those that end up being an important part of the story. But there's a lot of, what I want to leave you with this here, we're going to circle back, is there's a lot of selection going on across the genome here. Okay? So again, genome scans, this isn't any phenotype. We're just taking fin clips of fish from these sites and sequencing whole genomes. So, and like we said, these urban estuaries are polluted with lots of different chemicals. And these are highly disturbed environments in lots of dimensions, not just pollution, right? Presumably, flow regimes are really different, predators are different, pathogens, microbes, all these kinds of things are different in urbanized, highly altered, perturbed environments. And so what we're seeing here is sort of like the genomic outcome of selection for fitness in this highly altered environment with no real indication of what is the agent of selection here. So that's where we have started to go in our lab, is trying to piece apart and pull apart and try to figure out what's the complexity of adaptation in these environments, what are the traits that are involved, what are some of the selective drivers of this adaptive evolutionary change that's going on in these environments. So where we've started is by doing some quantitative genetics to map the genetic variation that is correlated with phenotypic variation in sensitivity to these chemicals. So this is the genetic variation that that we started with, right? So PCB resistance, in particular, phenotyping the severity of cardiovascular developmental malformations that are caused during development. Very low doses cause these kinds of screw-ups, okay? So progenitors from polluted site, clean site, F1 sibling, full sib crosses to give us F2 intercross mapping families, lots of embryos that we then expose embryos during development to a distinguishing dose of PCB 126. We phenotype those individuals. How severe are their cardiac malformations after a few days of exposure? Which we know is highly correlated with post-hatch survival. And then we genotype individuals across the genome and start to try to piece together what are the genetic correlates, what are the genetic predictors of this phenotypic variation and sensitivity. So any guesses in the room? Are we talking like a few loci of major effect or like a bunch of loci of minor effect? Few loci of major effect? Come on, brave person. One person in the back. How about many loci of minor effect? Oh, you're all wrong, except for the one person in the back. Sorry. Wonderful. Okay, so now you're paying attention. That's great. I know you will. So here we are, basically the strength of association. So going across the genome here, different chromosomes, 24 chromosomes. And the size of the peak is an indication of the strength of association with variation in sensitivity. And we've got this for each of our 4 populations, right? So we did this breeding design for each of our 4 populations, right? So really it's quite remarkable in this population. Those 2 loci bounded by the blue box and the orange box account for 70% of the phenotypic variation in our mapping family. And this population, those same 2 loci, 53%, 55%, 34%. So what we're facing here is few loci of pretty large effect, like accounting for a remarkable amount of phenotypic variation, right? So we contrast this, where we're looking at single chemical, single phenotype at a particular developmental stage, with this scenario of the signatures of selection across the genome. We've got a very different scenario here. Here we've got a lot of loci that are under selection. So what we did next is we started to sort of compare, like, our QTLs showing signatures of selection. How do we think through this? So we started with this locus on chromosome 2. And what we have in this figure here are the signatures of selection for that population and that region of the genome. So high FST, low reduced nucleotide diversity, so big footprint of selection in all of these 4 populations at this locus. But it's only a QTL in the top 3 populations, not in the red population. Red population, huge signature of selection there, no QTL for this phenotype. And we've got some other weirdness here, where we've got a QTL in all 4 populations, but not a big selective sweep at that locus in all those different populations. So very subtle signatures of selection, if at all, in only a couple of those populations. So QTL in all populations, but not a signature of selection in all, signature of selection in all, but not a QTL in all. So how do we think about this? And this is sort of getting back to that complexity of what's going to be happening in natural systems with complex environmental changes. So the way that we think about this is perhaps, this is consistent with perhaps there being multiple redundant mutations within the population resulting in a soft sweep. So we don't see the big signature of natural selection, but we see a strong QTL. And remember, the QTLs, we got single progenitors, so it's not a population-wide thing. We've got one resistant progenitor. We're selecting one sort of genotypic makeup of resistance that might not be representative if there are redundant mutations that are circulating within populations. And then when we think about signatures of selection in all populations, but not a QTL, this is a little bit easier to think about, right? Where the mutation is clearly adaptive for something, but for some trait other than PCB-induced cardiotoxicity resistance. Keeping in mind, right, that urban estuaries are multiple stressors, including multiple chemicals with diverse mechanisms of action. With that, Noah did a lot of the population genomics, Jeff did the quantitative genetics, and we're following up with a couple of students in the lab, Carolina and Kendall, with collaborators at the EPA and Woods Hole. So thanks, folks.
Hi, everyone. Thanks for coming. My name is Matt Sasaki. I'm wrapping up a postdoc at the University of Vermont right now. And today I'm going to talk about some work that I've been doing since I was a PhD student all the way through these postdocs on rapid adaptation to seasonal changes in the environment in these copepods. So we've seen multiple times already at this conference some version of this plot showing that the planet's getting warmer. This one is the sea surface temperature anomaly, which shows pretty clearly that over the last 40 years, the sort of average temperature of the ocean has increased pretty drastically. Naturally, that's concerning for many different reasons. But we also know from sort of a growing body of literature and studies that have been done in a wide range of taxa that populations have some capacity to adapt to these changes in temperature that might mitigate the impacts that we're concerned about. So if we want to sort of effectively and efficiently respond to these changes in climate, understanding how, when, where, why populations adapt to changes is kind of important. We also know, however, that populations experience variability in the thermal environment over multiple different temporal scales. So for example, this is climatology from sort of a typical estuary in the northeastern US. We have very cold waters in the winter, very warm waters in the summer. For longer-lived organisms like us, we just have to sort of bear that temperature range, right? We add extra layers in the winter, wear t-shirts in the summer. But for the vast majority of living things, this seasonal variation in temperature is experienced across generations, where one generation in the winter would experience cooler temperatures and they get progressively warmer as the seasons go on. This introduces other options for how populations might respond to rapid changes in the environment, including sort of genetic differentiation and the evolution of plasticity itself. And that's what a lot of my work is focused on. In order to look at this, we need a good model system. Copepods are super small crustaceans, super widespread, super abundant, in my unbiased opinion, the best model systems out there for whatever question you want to ask. The one that I focus on is this species, Acarcia. In addition to being widespread and abundant like most copepods, it also plays key ecological roles in some of these coastal communities and in biogeochemical cycles. And as an added bonus, it's fairly easy to bring this copepod in from the field to the laboratory environment and rear them for multiple generations under controlled conditions, making them an ideal system to look for patterns in adaptation over different spatial and temporal scales. So some of my other work has shown across this large latitudinal thermal gradient that populations of Acarcia are locally adapted. There's variation in thermal limits as well as thermal limit plasticity. But also doing this work in Connecticut, sitting there watching water temperatures fluctuate across this huge range, we started to wonder what's happening over those seasonal timescales. Right? Acarcia has short generation times, so experiences very different conditions across generations. And so that's what I'm going to talk about first. Is there seasonal adaptation in thermal limits, thermal limit plasticity, and some body size stuff in Acarcia? To look at this, we've been out and collected copepods a couple of times from Long Island Sound, focusing on sort of the warmest time of their seasonal occurrence, as well as the coolest here in December and November. We brought them back to the lab, reared them under common garden conditions for several generations to minimize previous environmental acclimation. And then we ran them through the sort of classic split brood experiment where we collected eggs from these lab stocks, split them into two groups, reared them at different temperatures, and then assayed phenotypes once they'd matured. The phenotype of interest was thermal tolerance, which we estimated as LD50, so the temperature where we saw 50% mortality in this population, and then body size of the adults. And this is what we found. So after these common garden split brood experiments, we saw some pretty stark differences between thermal limits in copepods from the summer as opposed to the fall, and drastic shifts in the plastic response of thermal tolerance to changes in developmental temperature. We saw something similar but a little bit less interesting with body size, where copepods from the summer were smaller, copepods from the fall were a little bit larger. There wasn't much going on with the plasticity, with the exception of this last, late November collection. And yeah, I'm happy to talk about what I think is going on there, but I want to move on to other stuff. For those of you interested in the molecular mechanisms that are going on here, I'm going to encourage you to go to Alison Hall's talk on Monday in the interplay between dispersal plasticity and adaptation section. She has some really cool transcriptomic data, and that lets me completely avoid talking about any of that today. So, like I mentioned, Acarcia is widespread. We saw in the lab that these lab-reared populations were exhibiting these large differences in thermal tolerance and plasticity. But we were curious as to what's happening across this latitudinal distribution. So here, sort of from Florida up to New Brunswick, we find tansas at various times throughout the year, and during those times, they experience these large seasonal temperatures ranges. Even down sort of in Florida, they're getting almost a 10-degree change in temperature. And so the second part of this talk that I want to go over, and what my current postdoc is focused on, is looking at whether or not we see seasonal changes in these phenotypes in natural populations, and whether or not the seasonality is evident across latitude. So to answer this question, we had to go out into the field, so visited each of these 8 sites multiple times throughout the year, and those collections covered sort of this large seasonal temperature range, catching sort of the time period early on in the season of occurrence, the peak season of abundance for acacia at each site, and then some time point towards the end of their seasonal occurrence, or late in the seasonal occurrence. With these collections, we measured individual thermal limits using critical thermal maxima sort of in the field. These are individuals acclimated to their field conditions rather than to laboratory conditions, and then preserved them for some low coverage whole genome stuff, which is still very much in the works, and so I'm not going to talk about that either today, and we'll focus just on the thermal limits. So this is what we saw. Perhaps unsurprisingly, within each collection, we're covering that large latitudinal gradient in thermal limits where copepods from the warmer southern sites have higher thermal limits than copepods from the cooler northern site or higher latitude sites. But the other thing that really stands out is that within sites, we see these large seasonal changes in thermal limits, similar to like what we saw in Long Island Sound with those laboratory experiments. And to put this all together, if you remember, we're kind of using seasonal variation in thermal limits and this rapid seasonal adaptation as a proxy for long-term change. And in order for that to be useful, we're really looking for something that aligns both the rapid seasonal variation in thermal limits with the long-term sort of latitudinal local adaptation. What we're seeing is that both within and across populations, a lot of the variation in thermal limits that we saw is explained by collection temperature, so the temperature they're experiencing. Because these weren't laboratory acclimated, this combines the effects of both plasticity and any sort of rapid genetic effects. That's something that we're hoping to get at with the low coverage genome data. To wrap up, the rapid adaptation we see over seasonal timescales has clear effects on variation in thermal limits, on body size, and in plasticity in these traits. That seasonal variation is widespread, reflecting some influence of both plasticity and genetic differentiation that we can start to dig into moving forward. And regardless of the mechanisms underlying this variation, plasticity, genetic differentiation, some combination of both, this seasonal variation in the environment is likely priming populations to respond to longer-term changes in the environment. So with that, before I take questions, I want to acknowledge the Pespini lab at UVM, the DOM lab at UConn for all of their help. And if you or someone you know are interested in these sort of spatial and temporal patterns in thermal adaptation and using that to look at ecological changes in the planktonic communities, I'm starting a lab at UMass Lowell in January, and I have funding and would very much enjoy talking to you about potential projects. And I will take questions now.
Thank you for the nice talk. So you showed like latitudinal gradient and like seasonal variations. And how about the daily variation? Is there also a pattern along the gradient?
Yeah, so daily variation with these copepods are tricky because these coastal systems are so tidal, and so they're getting admixed in and out. And so a lot of the temperature sensors that are stationary are picking up the changes in temperature driven by advection and overestimate the amount of variation experienced by these copepods. There's some modeling data out there which suggests that within the sort of advected water bodies, they're getting 1 or 2 degrees of daily temperature variation. Yeah.
It looked like for your different latitudinal populations, the sort of slope of the reaction norm is pretty consistent across populations. Did that surprise you? Like, does that mean that there are sort of like, no matter where you are in that curve, there are hard limits of like how flexible you are?
Yes, it was very surprising. In the sort of common garden experiments we've done across that latitudinal gradient, there is genetic variation in the slope of the reaction norm, similar to what we saw in the seasonal experiments, where warm adapted populations have shallower reaction norms. The way we're interpreting this is if you go in and actually pull out the slopes, the temperature sensitivity from these natural populations, there is slight variation, lower reaction norm slopes in the south. They peak in mid-latitudes, but then something in the natural environment in these natural populations is dampening plasticity in the northern populations. And so it's reducing what we saw in the field relative to what we know there's some genetic capacity for in the field. Excellent. Yeah.
Did you say there's discrete generations within that season?
Yeah, there are multiple potentially overlapping generations throughout the year. And so generation times are 2 to 3 weeks in these copepods.
So this is maybe a way out there idea, but is there any chance that heritability is kind of synced up across generations? So like it skips several generations through epigenetic effects or something. You see higher heritability between generations 1 and 4 than 1 and 2.
Yeah. There's some really cool work from other students in Melissa Pespini's lab that have shown sort of the changes in CT max after exposure to selection over time that suggests there might be some of those kind of changes too. Yeah. Okay, I'm going to hand it off if you want to get your slides ready.
Thank you, everyone. I want to start my talk by just acknowledging that the University of Rhode Island and a lot of the lands where we collected samples and do research for the projects I'm going to talk about today are indigenous lands, and I think it's important to recognize that. I also want to start by acknowledging basically my entire lab. I'm going to give you a brief overview of a few different projects, and this is a work of a small village and lots of hours of many different people, and so I want to express my gratitude to them right away at the beginning of this talk. So I sort of changed up, if you read the abstract or title of my talk, you notice that this is not what I had put in there, and I kind of decided to do a little taste of everything that's going on in the lab right now. So we borrowed from the local culture. I'm calling this a research amuse-bouche. And I also have some little sub-objectives today. I want to advertise and hype up some of the cool results from one of my PhD students who's going to finish pretty soon. I'm also hoping to recruit a postdoc soon for the lab, so hopefully I can inspire you to maybe want to come join us. And I'm also going to be on sabbatical in the fall, so if you want to learn more about any of these things, I'm happy to come give a talk. It's a good time for me to come and hang out. I'd love to do that. Okay, so, I was going to say, just like the bear, every second counts for this talk, so we're going to move pretty fast. And I'll start by giving you some of the take-home message. And the first one is that I'm going to show some experiments where we did larval expression or larval exposure studies, and I want to convince you that using multiple spawns to do some of these selection experiments are really important. The second take-home message is that I'm going to show you, using some of the same loci that we identified in our larval experiments, we were able to show that adaptive loci in adult populations are being shaped by similar coastal stressors that we've tested out. And lastly, I'm going to try to hope to convince you that looking across life history stages, or sort of what I just talked about, is really critical for understanding selection in marine species. So Andrew did a really good job of sort of introducing what a lot of coastal organisms face, but we can kind of build up this complex environment that they're living in. We have natural environment, urban environment, and this comes along with things like sewage effluent, different things coming in from roads and runoff. We also have agricultural runoff and livestock. All these things are interacting with marine species in the coastal environment, and I think the St. Lawrence River is a great example of this if you're driving in. So some of the main stressors that are present in these, and like Andrew said, there's a whole gamut of things that you could talk about in some of these habitats, but we focus in on a few different ones. We look at things like acidification, which is driven by the carbon dioxide, things like sewage effluent, low dissolved oxygen, also eutrophication. Talk about low salinity. We could talk about microplastics, PCBs, temperature. We've heard almost all of these things already mentioned in this session. So like all this stuff is going on. It's pretty crazy what coastal organisms have to deal with. And one of the things that we're really focused on is this pairing of coastal acidification and hypoxia. So lots of folks think about ocean acidification. You think about CO2 coming from the atmosphere, from different emission sources, and then being absorbed by the ocean and lowering the pH. But for a lot of coastal systems, especially in the northeast of the United States, where we're from, acidification, this coastal acidification, is actually driven by eutrophication, or the input of nutrients or fresh water into these estuarine systems. And basically, this can come from a variety of different sources. And when that nitrogen sort of increases, it drives microbial and community respiration, and that lowers the dissolved oxygen and increases the dissolved carbon dioxide, so we have low DO and low pH. And this has been shown to be very tightly mechanistically linked in a lot of different urban estuaries, where it fluctuates not only seasonally, but on a diurnal basis. So we're really interested in this diurnal fluctuation of these two stressors. We have a few different projects going on in the lab where we get really crazy and we pair these two stressors with a third stressor. Sometimes that's sewage effluent, sometimes that's salinity. But all of these projects really focus on looking across both or multiple life history stages in these marine organisms. So if you didn't recognize those little cartoons at the bottom, that's the eastern oyster. That's what we focus most of our research on. I'm not going to try to convince you how cool oysters are. Everybody knows all this stuff. Oysters are the best. But one of the things that you might not know is that oysters don't move around. They're basically like living rocks in the ocean as adults. But their larvae spend about 16 to 25 days, give or take, in the water column. And that's a really critical life history stage for these benthic organisms. So here you can kind of imagine two different oyster populations. They're basically entirely dependent on larvae for gene flow. So if everything's working well, we have larvae floating around from one population into another, and this is what's providing gene flow in those populations. However, these life history stages, these early ones, are very sensitive to different stressors. So if we suddenly are encountering these things that we introduced before, that might end up being a barrier to dispersal. But alternatively, we may have some larvae that are resistant to that, and they might be strong and to those stressors. And those are the ones that might be able to get through those barriers or those stressors that are in the water column. And so we're really interested in sort of taking those resistant phenotypes and figuring out where in the eastern oyster genome those SNPs are that are doing that. And so we do this in, I'm going to admit, a rather complex framework of how we do experiments and seascape genomics paired together. So really, I'm going to show you a couple different experiments today, and basically we keep coming up with really complex and distinct ways to torture oysters. It's usually early larvae, sometimes late larvae, sometimes juveniles, sometimes it's all of that. All these different stressors, so we focused a lot on the diurnal cycling, the sewage effluent, and salinity, as well as the low dissolved oxygen. And so we basically introduced those stressors to those early life history stages, hoping to see some sort of mortality. And then we try to sequence the ones that survive. And that helps us identify these loci with a consistent response to the stressor. So we're trying to look for allele frequencies that show a big shift after that. But in our control treatment, we're not seeing that shift. That's the ideal that we're trying to identify. And with that, we can get those loci that might be putatively under selection in early life history stages. We also can include random loci to do this. And we make capture probes. We do this using a method called EEG-seq, which I'll talk about in a second. So we get these probes from our early life history stages, and then we shift this over into an adult seascape genomics framework. So we take those same probes, and then we combine our adult DNA from them across these environmental gradients. And so hopefully we can sort of break this up into loci that are under selection in the early life history stage, those that might be under selection as adults, and neutral loci as well across these different environmental gradients. And we have sort of some different hypotheses of what kind of correlations or associations we might see depending on what we're talking about. And we've been using EEG-seq to do this. I'm not going to have a chance to really talk about this, but please go see Jacob Green's talk about that as soon as you can on Monday. I will start with a project that I began as a postdoc that was sort of the inspiration for a lot of this work. We focused on coastal acidification and sewage effluent, and that's because these two things are highly linked to each other. You can see that hotspot of acidification there by a sewage treatment plant. And if you weren't aware, sewage effluent is everywhere in the marine environment. We have crazy levels of volume, billions of liters every day in many of our different urban estuaries. It's filled with different contaminants and things that come through. Any one of these could really induce larval mortality, and we know that the wastewater and the acidification are tightly linked. So this was our first crazy torture experiment. We did mass spawning. We had a whole group of larvae. We took initial genomic samples from there, and then we split them up into 4 different treatments: a coastal acidification treatment of about 2,800 microatmospheres, a 5% sewage effluent treatment, the 2 of those things combined, and then a control. After 24 hours, we pooled and then used the survivors for this exome capture to try to understand these loci. And we did this across 3 different replicate blocks, which is really critical. And so one of the take-home things we found was that across the blocks, we saw a similar response where adding sewage effluent really increased mortality. So this was one of our major drivers. And there was a little bit of an additive effect, but it wasn't significant between our multi-stressor and the single sewage effluent one. We were able to identify about 2,000 different outlier SNPs across 481 genes. And I'm going to show you a PCA result of that from across all of our spawns. So if I plot them all like this in PC space, you can see that there's not really a great pattern. You're like, wait, I don't see any treatment-specific effects here. And that's really because all you're seeing is the different spawns together. So we have really these, they're related to each other. And so it's bringing up this really independent pool of genomic loci. So it's really important to incorporate this because it's really clearly different. However, even though we have these different spawns, we have a consistent pattern in what happens. So the initial samples are in that blue, the control are in gray, and then we have our acidification treatment, and then the addition of that sewage effluent sort of models the mortality where we see this split between the multi-stressor and the single stressor with the sewage effluent. And this is consistent in 2 of our blocks and largely consistent in a 3rd one, though it's slightly different. But I put it at the bottom so most of you can't see it, so don't worry about it. But the other really cool pattern of using these multiple spawns is that if you just looked at outliers from one of the spawns, you got this really kind of just stressor-specific pattern that didn't really make sense. But if you limit an outlier to having to show up in more than one spawn, suddenly we have this pattern where we see our multi-stressor is holding a lot of shared outliers with our single stressors, which makes perfect sense. It's a multi-stressor. It's a linear combination of the 2. So we finally got some things that made sense when we looked across these multiple spawns. So again, I hope I've convinced you with that brief introduction of how important the multiple spawns are. So now I just want to highlight some work of a project that PhD candidate Amy Zick led. This is where we added in a 3rd stressor for a project and did some crazy exposures. I'm not going to talk about the exposures because I want to focus on the adult samples, because this was really our first time where we got to link the adults and the larvae together. And if you missed Amy's talk yesterday, the main result takeaway was that she identified that basically multiple environmental variables, including salinity, temperature, dissolved oxygen, and pH, are driving population genetic structure in these adults in Narragansett Bay. And so this is a really cool result. I'm just giving you a taste to whet your appetite, but really, I want you to talk to Amy about it. She has a great repository set up with the analysis, and she's graduating in December, hint, hint. So I just want to make sure everyone's aware of that. So we're doing this in Narragansett Bay, but we're also expanding across multiple urbanized estuaries that's coming really soon. So I'll move on to our last and 3rd point since I'm running out of time. I can't even begin to describe the multiple larval and juvenile exposures that we have for this project, but this is involving cycling pH, cycling dissolved oxygen, and salinity. I'm just going to focus on what we tortured the juveniles with. So these were newly settled juveniles, and they were tortured with these diurnal cycles of pH and dissolved oxygen, and a weekly pulse of low salinity going from 30 to about 5 parts per thousand for 48 hours. And this was supposed to run for 4 weeks. We wanted to get 50% mortality. It ran for 90 days. And thanks to hard work from a postdoc and several students in the lab, we finally got some results 90 days later. And what we found was that the salinity pulse was really important in driving mortality for these early juveniles, which was a little bit of a surprise. And we didn't really see an impact between combining those stressors together. The other really interesting thing that popped out is that growth-wise, the cycling acidification dissolved oxygen treatment did the best in terms of growth. Though I have some hypotheses about this. That was based on an image analysis, and I think some of the shell is not real shell that we're counting in a photo, but we're going to be adding these really cool 3D x-rays of the juvenile shells to help gauge the actual density. That coloring there is actually showing the density of the shell as it's moving in 3D space. And now that I'm out of time, I can come back to my 3 take-home messages, but I also leave you with dessert. And that dessert is to hire Amy, come talk to me about doing a postdoc in the lab, and I'd love to come give a talk at your university next semester. So with that, I'll thank you all for coming.
So to motivate a little bit about my talk, as we know, we are facing a biodiversity crisis where we are seeing declines in population sizes all over the world. And this is mainly a result of change in land usage and also the climate crisis. So to think about my project, let's think about a little bit of why the changes in climate are producing a decrease in populations. So as we know, species are locally adapted to their environment. So this means that species are in match with the environments they are adapted to. But when we see climate changing, we are seeing that now species are facing a mismatch in between their distributions and the climates in those current distributions. So when we are seeing this happening, there are only 3 possible outcomes. The first one is that the populations migrate or shift their geographical distribution to follow optimal climate. The second one is that populations might adapt to the newly imposed conditions, or the third one, and the one that we want to avoid, is that populations might face local or global extinction. So my lab and I particularly are focusing on the second strategy, and we are asking, can populations and how are they going to adapt to the newly imposed conditions? Can this happen? So when we think about this, we might be thinking about the fact, is rapid adaptation predictable? And one key factor about rapid adaptation, so something that I wanted to say here is that we're not only talking about adaptation because changes in climate are happening very quickly. So we need not only adaptation, but rapid adaptation. So because we're talking about rapid adaptation, and this is gonna happen in not many generations, but short periods of time or not that many generations, we are talking about adaptation acting on standing genetic variation rather than acting on, for example, de novo mutations. And the fact of how this standing genetic variation is distributed across the genome maybe is going to have a high effect on if populations are going to survive or not. So we are thinking about how does genetic architecture affect the probability of populations rapidly adapting to a novel environment. And I'm going to try to answer these questions through genetic simulations. So first, I'm going to be walking you through from the setup of my simulations. So because we were interested in the effect of genetic architecture on populations' probability to survive in a novel environment, we decided to model very different situations for polygenicity because this is a key parameter in genetic architecture. We simulated an adaptive trait made of one causal locus to thousands of causal loci. We also modeled 5 different heritability levels, and we also took alleles from the side frequency spectrum from a real Arabidopsis thaliana population that I'm going to speak about later. I'm not going to go through all of this because I don't have time, but we use classical quantitative genetic equations to build our trait. But I just want to mention, if any of you are interested in this, we actually ran many different simulations, changing the distribution of effect size from normal distribution, beta distribution, making them dependent on the allele frequencies, independent of the allele frequencies. And then we use an additive model in quantitative genetics, and then with that, we model our phenotypes. Once we had our phenotypes, we apply a Gaussian function to model stabilizing selection. as you can see here. And then, to make sure the comparison in between different genetic architecture was fair, what we did is we started all the simulations across all genetic architectures with the same phenotypic mean and variance. Also, because we were interested in seeing what were the effects on genetic architectures on the probability of populations to adapt to a new environment, handle a small environmental change or a big environmental change, we modeled many different environments. So from close by environments to faraway environments, thinking about small or big changes in the environmental conditions. Also, because we wanted to set up these simulations not only to understand general patterns, but also to see if we could inform real-world experiments, we set up these simulations resembling a real-world experiment that our lab has been leading that is called GreenNet. My advisor is going to explain this experiment, so you should go on Monday. So we also use many different characteristics of Arabidopsis thaliana. We use real genetic data from 231 ecotypes, reproductive strategies, recombination rate, non-overlapping generations, and also we fitted the carrying capacity and offspring size from real experiments. We conducted the simulations in SLIM, and we ran the simulations only for 10 generations because we were interested in seeing how genetic architecture could affect survival of populations after a little period of time. So, okay. So these are the results that we got. So first, let's see how heritability and the size of the environmental change affected survival. So in this plot, I'm showing in the X the different levels of heritability from low to high, and in the Y the environmental change from low to high. And as it is expected in each of the boxes, I'm showing the percentage of survival of the populations. And as you might expect, populations with low heritability and a huge environmental change in most of the cases died. And populations with high heritability and small environmental change almost always survive. But because we were interested in the relationship between genetic architecture, and one key parameter of genetic architecture is polygenicity, once we observed this parameter space, we were asking in each of these little boxes what is going on in between survival and polygenicity. So we ran multiple linear regressions to ask what is the relationship in between these two, polygenicity and population survival. So we can say if we saw a positive slope in the logistic regressions that high polygenic architectures have higher survival, and negative slopes indicate that low polygenic architectures might have a higher survival. So here in this plot, I'm showing you the same parameter space, but now the boxes, instead of being indicating survival, they are indicating if it is beneficial to have like a high polygenic trait or not. And we were very surprised because we saw these two different scenarios. We saw that in close by environment, high polygenicity seems to be good for survival of the populations. And in faraway environments, we are seeing that low polygenicity seems to be more beneficial for the survival of the populations. So we were very intrigued about these results, and we were like, what is going on here? Because if we started all the populations with any genetic architecture leading to the same mean and variance in the phenotypes, why we are seeing these differences in survival? So then we started asking what could be the effect of genetic architecture on fitness variance. As Fisher said many decades ago, is the probability of the populations to evolve. So in this next plot, I'm going to show you the two different scenarios that we observe: small environmental change, big environmental change, and then I'm going to be showing you survivalship, fitness variance, and then the different dots are going to be colored by the polygenicity of the populations. So populations with low polygenicity are going to be darker, and populations with high polygenicity are going to be lighter. So let's see. So here, the first pattern that we see is that under small environmental changes and under big environmental changes, we see a positive relationship between fitness variance and survivalship. And this was expected. Populations that have higher fitness variance have a higher probability to survive. And then we also see the results that we saw before, where under small environmental change, we see that all the high polygenicity populations survive because this is very light, and under big environmental change, the only populations that survive are the low polygenicity ones. But we see something interesting, and it's the distribution of fitness variance across the different polygenicity levels. So we see that high polygenic architectures in small environmental change or big environmental change tend to cluster around like a very narrow set of values. Here, here, and here. But low polygenicity architectures, even though here don't show a higher survival and here are the only ones that survive, have very wide range of values of fitness variance. So why is that? So instead of walking you through more and more plots, I'm gonna show you what we found in cartoons because it's easier to understand. So we know that low polygenic architectures and high polygenic architectures reach very different distribution of phenotypes. And this was described by Fisher a long time ago. We see that low polygenic architecture have more discrete distributions with values of the different phenotypes being very dependent on the initial allele frequency and the effect size of the contributing alleles. While high polygenic architectures tend to be more continuous and resemble a Gaussian distribution. So this was described, but now what is the effect on this distribution of phenotypes on the survival of the populations when they are submitted to certain selection forces? So in our simulations, when we put the populations under small environmental changes, what we see, and here I'm describing the Gaussian force of selection, the Gaussian distribution of natural selection, what we are seeing is that because low polygenic architectures have more like discrete distributions, highly depending on initial parameters, they reach higher noise of fitness variance. While high polygenic architectures, because they tend to a more continuous distribution, they have a lower noise in fitness variance. That is what I showed before. And then when we submitting the populations to big environmental changes, because of even though the starting conditions of the simulations where all the populations have the same mean and variance in phenotypes, because of the nature of low polygenic architectures, they have more extreme values. So then when we have a more extreme change in the environment, the only populations that can make it are low polygenic ones. While for high polygenic architectures, because they follow the trends of a Gaussian distribution, when the new environment is outside their continuous distribution, they just don't survive. So the overall take-home message is that under our assumptions of our simulations, and when populations only depend on standard genetic variation, we see that low polygenic architectures are more unpredictable, but we survive in small environmental change and big environmental change, while high polygenic architectures have more predictable results of survival and mortality in small and big environmental change, respectively. So one question is, how can we think about these results in real-world experiments? And this is what we're doing right now, because we have this GreenNet project that is a huge project of multiple environments, multiple years, where we have the same population of plants planted in very different environments, in harsher to less harsh environments, and we want to see if we can see differences in the genetic architecture of adaptive traits in these different environments. So this is what I'm working right now, and keep tuned if you want to hear the results in the next evolution talk. So with that, I want to thank my lab for very useful feedback in this project, and I will leave you with other talks from my lab mates, and I would love to hear some questions, or if you want to come to me if you have questions about the simulations or comments or critiques or whatever.
Describe these extreme values at the end of the distribution for lowly polygenic traits as noise. Do you expect those to be the similar sets of loci that are on the extreme end there, or would you expect to see higher repeatability in evolution with lower polygenic traits and higher distance reality?
Yeah, exactly. So we expect to see more repeatability or predictability but for low conditionicity, we should see that if you get to a checkpoint that you're like exactly in the new environment, then you do great. But if you're not, because the effect size or the initial frequency of the contributing alleles was not the right one, then you're